Uh, so some things to consider in preparing the DMF. Your manufacturing supply chain, this is going to go into your 32S21 section. You need to specify all the sites that are involved in your supply chain. So if you're using an outside lab to even test residual solvents in a raw material, you need to list that in your 32S21 section. And sometimes, I don't think it happens so much with drug substances, but with drug products, sometimes the supply chain folks are waiting to make their decision, and you're like, I need to know this because I need to finish this section. I don't know if anyone here has found that to be a problem with the drug substance supply chain, but certainly we've had issues with drug product supply chain. Your raw material supply, this is all going to go into your 32S2, and there's a subsection 3, control materials section. Here I stated we don't need to commit to raw material suppliers unless it's a unique starting material, but you're going to need to commit to the controls. Now on the checklist for this completeness assessment, I did see checkbox, have they identified the source of the starting material? So I think that that is changing. That they're not just comfortable with, here's my starting material, here's the specification. As long as it meets this, it doesn't matter where we get it. The concern is that if you get it somewhere else, are there new impurities that you're not necessarily looking for, or they want to make sure that you're looking for those impurities if it's a different source. So I see that changing. And certainly as we go into Europe, they are very keen on starting materials and identifying the starting material suppliers. You need to commit to your controls, not necessarily everything that you're doing incoming. And I try to strike a balance between too much information and enough information. And I'll get to that a little bit later. I wanted to talk about how maybe you could populate your DMF, and I'll get into that a little bit later. Process validation, again, it's not required. Not typically required for an API. You don't need to have your process validation complete before you file your DMF, but you need to be comfortable with the in-process controls that you've provided, the specifications that you've provided. If you go to scale, if you haven't done any scale up, you don't have any full-scale data, you don't know what's going to happen when you do scale up. Are you going to all of a sudden, we need a tightly wider spec on that impurity, or that in-process control, or I need to add an in-process control, or my intermediate spec needs to change a little bit. So even though it's not required that you've conducted your process validation, you do want a level of comfort with your process when you're preparing your DMF. If you have to change things, these are amendments to the DMF, and you'll need to notify your customer, and they may have reporting requirements that may delay them being able to use this material. A synthesis development report. Some of this information could go into 32S26, your process development. Really, you want to focus on major changes that happen during your development of your final API process, particularly with respect to the impurity profile. They want to make sure that you're not introducing new impurities or that you were trying to actually clean things up. Some of the information from a synthesis development report may go into other sections. Just if you had polymorph screenings, and it may go into a different section, a justification type of section, or a in the 32S21 section. More, this is the characteristics of our API. Impurity identification, important, and I reiterated here, pay special attention to potential genotoxic impurities, and I'm sure everyone's probably pretty aware of, there's the ICH guidance around impurities. So if your drug is being used in a drug product with the dose is less than 2 grams a day, you're going to need to be able to identify any impurity that's greater than 0.10%. And if it's greater than 0.15%, you're going to need to be able to qualify that impurity. If the drug product that it's being used in is greater than 2 grams a day, those levels drop to 0.05%. So you need to be conscious of those those impurities that you're seeing in the levels, and you need to be able to justify that your levels are considered safe. And this is another reason, why the FDA needs to know what final product are you using this in. You may have some impurities that would be appropriate for one drug product, but not for another one. Analytical development and validation, you need to summarize all your validation activities. And again, even if you're using a compendial chromatographic method, you need to do some kind of verification in your labs, and they do want to see that. Particularly the generic division does want to see that information. You'll need to develop an internal stability protocol and study. Usually 3 lots at accelerated and long-term conditions. I'm sure everyone's familiar with ICH conditions. It's very prescribed. I have done DMFs where we've provided data on pilot scale lots, but again, you're going to need to commit to doing full-scale lots. So having said that, I just wanted to comment. I don't have this on a slide. There are different ways to actually prepare your DMF. Some may prefer to submit internal specifications, internal methods, so the actual methods that your chemists are using in the lab to perform the analytical methods. Some may want to submit a complete development report. What I like to try to do is, in terms of the complete development report, I said some of that information may belong actually in another section. I like to try to give the FDA a story instead of having them try to figure out and meander around, because development is development, and you may have to go off and figure something out that happened, and it might be a little bit of a gap. It might be hard to read through that as an FDA reviewer. So I like to try to tell the story, not hide anything, but tell the story in a manner that is easy for them to review. And certainly you can also link to internal reports if you're more comfortable doing that. But I like to tell the story. And in terms of analytical procedures, I prefer to have registration type of documents where it's more on the level of how a USP method is described. This is the concentration that we want of the standard and the sample. But I don't want to tell you I'm going to take 10 milligrams, put it in a 50 ml volumetric flask, stir it for 30 minutes. I just want to tell you I'm dissolving it in acetone, and this is the final concentration. There are different schools of thought on that. I think it's much easier to maintain your DMF if you've more told a story and if you are giving yourself some flexibility in the lab. If you are submitting internal laboratory reports, every time that changes, you're going to need to amend your DMF and put that new analytical method in there. Even if it's a typo, even if you've just reformatted all of your internal documents, I think it's just much harder to upkeep the DMF. Even though the changes aren't significant, it's still an activity that needs to happen. And if you have summarized your analytical methods, changes could happen, and that's not your commitment in your DMF. So you're, I think, a lot better off. But it does take some upfront work to do that summarizing, to do that paraphrasing of the analytical methods. In my opinion, it's worth the effort.